Hi everyone, for this lecture we'll be talking about Fundamental Research Issues, Chapter 4. In research, we are measuring variables, how they relate, how they influence or are influenced by something. But what is a variable? Really, a variable is just something or anything that changes. What are some variables you know that are measured in psychology? This can be behavior, thoughts, feelings, temperature, size, and so many other things. Anything that varies and can be measured is a variable. Some variables have an obvious way of being measured. If your variable is length, you measure the length with a ruler or other measurement device. But if your variable is an emotion, it isn't always obvious of how to measure it. Which brings us to operational definitions, which are a precise description of how the variables in a study will be measured or manipulated. How would you measure memory, personality, emotion, cognition, intelligence, or other variables that aren't always visible or easy to measure? Having an operational definition is key in making psychology an empirical science because it forces scientists to discuss abstract concepts in concrete terms. And without it, we wouldn't have a way of talking about the variables in a systematic or experimental way, and we wouldn't be able to replicate the research. For instance, if we are talking about aggressive behavior, what counts as aggressive to you? Is it physical? Is it verbal? Is it both? And sometimes people tease each other or slap each other on the back in a friendly way. So context also matters here and needs to be defined. If you just say aggression and one person measures it verbally and the other physically, you can't really compare your findings and we can't replicate your research. The non-experimental method is also known as a correlational method. And as you know from correlations, this method has measurements of variables to determine whether variables are related to one another as they occur naturally. So you're not manipulating the variable in any way. We can also look at descriptive research, which is observing and describing behavior, again, as it occurs naturally. We aren't changing anything. This might be done through, say, observation or going back to look at census data. But there are problems with non-experimental research. First, it cannot determine cause and effect. Because nothing is being manipulated, we can't say that variable A causes variable B. For instance, if we find that there is a positive correlation between class attendance and test scores, we can't just say that coming to class increases your scores on the test. It might be that students who do well on a test are just more likely to come to class. We might also have a third variable problem, where another variable might be influencing this relation. You may have heard that there's a positive relation between ice cream sales and sunburns, but we know that ice cream sales don't cause sunburns, right? Rather, a third variable, the sun, is influencing this relation. We also have to worry about extraneous and confounding variables. Extraneous variables are variables in a study other than the variables being measured, while confounding variables are variables that are not controlled for in a research investigation. We try to control for both types of variables, making sure that we are measuring as many things as we can, but it's not always possible. There can always be other things influencing the data, especially in non-experimental research, and especially in human non-experimental research. But non-experimental research isn't all bad. It's a great starting point for future questions. If you see a relationship that's surprising, it can lead to all sorts of new research questions. It can allow for meaningful predictions. Again, we can't say that there's a cause and effect here, but we can say that there is a relationship. And if there's a strong positive association with, say, attending class and test scores, we can predict how many people will likely do well on a test based on their attendance. We can also use non-experimental research when we cannot or should not manipulate variables due to research ethics. If we want to examine the relation between smoking while pregnant and negative outcomes at birth, we can't assign pregnant women to be in an experimental group in which they need to smoke cigarettes. But we can look at those who did smoke while pregnant and then look at outcomes at birth associated with the smoking status. The experimental method is a method of determining a cause and effect relationship. In a true experimental method, a researcher manipulates the independent variable, measures a change in the dependent variable, and controls all other variables by either randomization or direct experimental control. So let's break this down a little bit more. An independent variable is a variable that is manipulated to observe its effect on the dependent variable. It is a thing that you are changing, adding in, removing, or manipulating in some way. The dependent variable is a variable being measured in a study. I like to think about it as being dependent on the independent variable. Some examples here. If a farmer is wondering about the effect of a fertilizer on plant height, they can try different types of fertilizer. Because this is something you can manipulate, it is the independent variable. Plant height is hypothesized to depend on the fertilizer and is the dependent variable. Similarly, if a researcher is wondering about strategy use on working memory, we can assign a participant to one of two strategy conditions, making it the independent variable. Because working memory is the outcome and hypothesized to be dependent on the strategy, it is the dependent variable. Lastly, if a researcher is interested in how different drug doses affect the progression of a disease, we can have different types of drugs, which is the independent variable, and then measure disease progression, the dependent variable. The second part of the experimental method is experimental control, which is eliminating the influence of extraneous variables on the outcome of an experiment by keeping the variable constant in the experimental and control groups. So we typically do research in a laboratory setting because it helps us to control as many variables as possible. We can control temperature, noise level, how many people are in the room, and so on. Having control over the environment allows us to know that these variables did not affect the results. But there's always going to be things that we cannot control. For instance, if we are interested in depression, but our study is collecting data in the spring and summer, we are not accounting for winter seasonal depression. If this is something that we know might affect the data, then when we go to write up the results, we would put this in the limitation section and state that in the publication. Another important component is randomization, which is controlling for the effects of some extraneous variables by ensuring that the variables operate in a manner determined by chance. This is really important in science, especially given that there is an impossible amount of extraneous variables to control for. Say a participant is stressed, tired, having relationship problems, is happy. All of these might affect how someone performs in the lab. And by having randomization, it makes it so extraneous variables are likely to affect all conditions or groups equally. It can include what order stimuli are presented in, the time of day the participant comes in, and other such examples. One really important method of randomization is random assignment to condition, where we randomize whether participants are in the control or experimental group. You can imagine that if we are hypothesizing that female participants will have more sensitivity to a mindfulness treatment than males, we don't want to have female participants come in and then we say, oh, we should put you in this mindfulness group, because that will affect the results. And sometimes we do this consciously, wanting to influence the outcome, but other times researchers might do this without even knowing that they're doing this. So instead, we tend to use something like a random number generator or coin toss to determine the group. We also need to worry about validity, or the extent to which a conclusion is reasonably accurate. We evaluate the validity of both our research and that of others, and we do that by breaking it down into different categories. Some we will talk about now, and others we will talk about later. Construct validity refers to the extent to which the measurement of manipulation of a variable is accurate and represents the theoretical variable being studied. Is the way I measure intelligence a valid measure of intelligence? And we can examine this by asking ourselves if the operational definition reflects the true meaning of the variable. If I give a questionnaire of people's opinions on different fruits and say it's an IQ test, it probably doesn't have good construct validity because that isn't my definition of intelligence to start with. We measure construct validity by seeing if it is correlated to other things that it should be correlated to. If we are measuring intelligence, is it related to education level, income, occupation? If not, we might not be getting at intelligence. Next is internal validity, or the ability to draw conclusions about causal relationships from the results of a study. This is essentially being able to say that A causes B, meaning that it's also limited to true experiments. And to have internal validity, we need three things. First is temporal precedence, which is the idea that the causal variable should come first in temporal order of events and be followed by the effect. So if stress causes illness, stress needs to be present before the illness. A needs to come before B if A causes B. Next is covariation, which is observing that change in one variable is accompanied by change in a second variable. So stress is causing illness. If stress is reduced, that means that illness should be reduced too, and vice versa. As one changes, another changes. And then we have eliminating alternative explanations, which is getting rid of extraneous variables. This could be getting rid of or statistically controlling for lack of sleep or nutrition, exposure to viruses, or anything of that nature. The next type of validity is external validity, or the extent to which the results can be generalized to other populations and settings. If you find it in a lab, can you find it outside of the lab? If you find it in college students, does it apply to high school students? Essentially, this is looking to see if it fits in other settings. This is why replication is so important in research. Lastly, there is statistical validity, which examines the accuracy of the statistical conclusions drawn from the results of a research investigation. This is essentially using statistics appropriately. Do we meet the assumptions of the test being used? Did we use the appropriate test? And did we interpret the findings accurately? So the activity for this chapter is on testing hypotheses. You will read each of the following hypotheses, and for each of the following, so you'll do this twice, answer the following four questions. 1. What type of method should be used to test a hypothesis, non-experimental/correlational, or experimental, and why? 2. Should the labels of IV, or independent variable, and DV, dependent variable, be used to describe the variables in the study? If these labels should be used, identify the variables and whether each is an independent or dependent variable. And so as a reminder, independent and dependent variables are only used when your study is experimental. These are not used for non-experimental or correlational studies. 3. Develop operational definitions for both variables. Remember to make these as specific as possible. Someone else should be able to read your definition and be able to measure or manipulate the variable in the same way. And then 4, describe the relationship that is expected to exist between these variables. And that's all I have for you for this chapter. Please let me know if you have any questions.